right, now we're recording. So while we're waiting, does anybody have any questions about anything? Well, that makes it easy. I guess maybe I'll go ahead and do some relatively unimportant announcements and reminders. I say relatively because I'll probably either be repeating myself or the information's not that important anyway, because I'd prefer to wait until after 8 before I do anything, share anything too important. Let's see. I guess the first thing I want to do is thank you guys again for letting me teach online on this Friday. Had some family stuff going on like I did last Friday, where I knew I would be cutting it close to getting to campus on time. So it was definitely possible for me to teach on campus, but it was also very possible that it could have gone south and I could have been late. So I was just planning ahead, so I was not late, and here we are. So thank you for that. But again, like I said in the announcements, when I told you that we would be online, like I said, it's all up to you guys. If anybody ever objects, if I say I'm going to be online and you don't want to be online, then just let me know, because I'm here for you. You know, you're the customer, so to speak. So in the future, make sure you let me know. On a related note, because not only did I have that family thing going on, but I have a lot of stuff going on with work and with travel. So normally my office hours are from 9 to 10:40. Today I have a meeting. I have a department meeting that starts at 10. It should be over by 11:30. So from 9 until about, I don't know, 9:50, I will be online as usual for office hours. And then from 10 to about 10:30, I'll be unavailable because I'll be in the biology department meeting. And then after 11:30, you should be able to reach me via phone. So if you call my office phone, 304-766-3125, it's at the bottom of my email signature, and in the syllabus, then you can just call me. Like I said, it's forwarded to my cell phone. So if you call my office, you'll reach me. I will be in my vehicle. So I won't be able to look at a computer or anything, but I will be able to talk to you if you have any questions. What other announcements? Next week is spring break. Don't show up unless, you could show up, but you might be by yourself. But despite the fact that it's spring break, like I keep saying, I'm here if you need me. If you want to use this week to get caught up, it's a good week to do it. Because I'm not taking the week off. So don't feel like you're bothering me if you ask me to help you over the week. What else for announcements? I think that's it. Office hours, spring break, meetings. Yeah, that's it. Perfect timing too, because it's 8. So we'll go ahead and get started. For attendance, if you're watching this video, as opposed to you guys who are here live, I want to remind you, the first question or the second question, because the first question asks your name. The second question asks you, it's a 3-part question. Well, let me think. Hold on, let me retract that. First of all, you need to say whether you're watching for attendance or for independent work or any other thing. Usually people get that part down. Then the next part is the 3-part question. You need to do the day and the date. Wait, no, 2 parts. Sorry. The day and the date. So say something like, I am writing for attendance for Friday, March 18th. And I know that sounds nitpicky, but it's a free point. And a lot of some of you guys are losing the points. And it's one of those things. I told you at the beginning of the semester, the purpose of, in my opinion, the purpose of a bachelor's degree is not necessarily to prepare you for a job. That's one of the things, but it's to make you a well-rounded citizen, a critical thinker. But I will admit that, yes, one of the purposes of a bachelor's degree is to prepare you for the real life. So, following details or following instructions and attention to detail is one of those things. So please pay attention when you do the questions. And for those of you online, right now, Schmidt, Knox, Vance, Flores, White, and Cordone, it's just attendance as usual. Just send me the email sometime before 9 a.m. And the first word for attendance on that note will be book, like a textbook. or a checkbook, or a book that you read for pleasure? Any questions? All right, let me pull up the PowerPoint. Oh, I should have had this pulled up earlier. My apologies. Oh, and Hamrick joined us. Cool. Good timing, Hamrick. Hamrick, you did miss the announcement, so if you get a chance after I post the video, you might want to go back and take a look. Oh, that reminds me. That was the other announcement. Since I will be traveling, it will be a while before I get that video and the questions posted today. So if you know anybody who is not online right now and who will be looking for the video, you might want to give them a heads up, send them a text, or let them know. Like I said, if you know somebody. Anyway, here's where we are. Can some wait? Not yet. There we go. Can someone please confirm that you can hear me and see the normal PowerPoint?
Yes.
Perfect. Thank you so much. All right, so here's where we ended off. Let me back it up a little bit really quickly. We were talking about what Mendel was up to in his little garden. And we talked about, we started this main bullet point of Mendel's law of segregation, but we never actually got to it. All we did was talk about this stuff about the monohybrid crosses, which is basically, the prelude, the information you need to know to understand his law of segregation. So now that we're done with that, we're going to get into it. I'll remind you what we talked about with the law of segregation was basically this. Again, he did this thousands of times. Purebred purple flowers and purebred white flowers. He bred them together. And every single time, 100% of the time, after thousands of times of trying, he kept getting a purple flower. And, obviously that was kind of weird. He's like, what? That is weird. Is the gene for white flower lost? And of course, again, they didn't call it genes. And also we know it's actually an allele, not a gene. But that was the question, right? Was the thing responsible for the white flower lost? So then after those experiments, he bred these. Because remember, this is a simplification. It's not like he crossed this one plant with this one plant and then you had one plant. No, there was hundreds, thousands maybe even of these purple plants, right? It wasn't just one. So anyway, he had this F1 generation. So maybe he crossed this purple one with this purple one. Of course, you can't tell it's purple, but anyway, he crossed those. And he did that thousands of times, all these different plants from the F1 generation. And this time, what he found was that there were some white ones. Specifically, about 25% of the time, they were white, and then 75% of the time, they were purple. That was the general ratio. It doesn't always have to be exactly 25% and 75%, but it's close because this is all a game of chance, as we'll talk about later. So anyway, that's where we left off. That's what he came to figure out with the monohybrid crosses. He figured out that the F1 plants that were all purple, despite the fact that they were purple, they did still carry, it said genes in the book, but we know it's an allele, but an allele for the white and an allele for the purple. So that allele for the white, it didn't disappear. It was just masked by the purple. So that brings us to where we left off. We are now caught up. All right. From the results of those experiments, he came up with 4 hypotheses. The first one was there are alternative versions of genes that account for variations in inherited characters. Now, of course, again, he didn't call them genes, so this isn't verbatim, but he did figure out from those experiments, or at least formed a hypothesis from those experiments, that there was something responsible for flower color in that example. And remember, there was a bunch of other things he did too, but using the flowers as an example, he figured, all right, there's obviously something responsible for the flower color, and there's 2 different versions of it. There's a purple version and there's a white version. That was the first one. The second, like I said, the alternative versions, we know them now as alleles. The second hypothesis that he came up with as a result of his experiments was for each inherited character, an organism inherits 2 alleles, one from each parent. And you already know this because you know this, at least from the chromosome level. You know that your chromosomes, right? You got one of each chromosome from each parents. You learned that in the last chapter. And then if you extrapolate that data and think to yourself, well, okay, genes are found on chromosomes, So that would make sense, right? That you would have one allele from one parent, one from another, because you have one on one chromosome and one on the other. So yeah. Any questions so far? Okay. As you already know from lab, hopefully this part is a complete reminder or refresher for you. An organism that has two of the same alleles is homozygous. So for example, if you have blue eyes, you have two recessive alleles. So specifically, you'd be homozygous recessive. If you have, well, let's just leave it at that for now. An organism that has two different alleles is called heterozygous. And as I mentioned on Wednesday, and I failed to mention on Monday in lab, think about these prefixes. You guys know these prefixes. Usually you hear these prefixes in the context of sexuality, right? A heterosexual is somebody who's attracted to other sexes. A homosexual is someone who's attracted to the same sex, right? So hetero is different, right? And homo is same, basically. So hopefully that'll help you remember that for the exam. Any questions so far? All right. Here's a little meme I found. I did not create. The heterozygotes, just a little uneven. I enjoyed it. I don't necessarily expect you to. Number 3, because remember he had 4 hypotheses that he came up with. If the two alleles are different, again, we're talking about a heterozygous condition here. If the two alleles are different, then one of them determines the appearance and the other one's basically useless. And the one that determines appearance is what we call dominant, and the one that is basically useless is what we call recessive. So again, with those purple flowers, when you cross the purebred purple flowers with the purebred white flowers, remember, none of those were this situation. They didn't have different alleles. Those were purebred. But all of their offspring obviously were purebred, and they were all purple because purple was dominant to white, which was recessive. It's pretty simple stuff, right? The dominant one you see, the recessive one you don't. Any questions? All right, let us move forward. Oh yeah, sorry, this stuff too. You already know this as well. When we are writing alleles or genes, if you will, depending on how you want to say it, we use uppercase to represent dominant and lowercase to represent recessive. So if you are big P in this situation, you are homozygous. If you are big P, little p, you are heterozygous. If you are little p, you are homozygous. Specifically, you are homozygous recessive. And of course here, like I said, you are heterozygous. And here, you are homozygous dominant. Now, I think this is simple, and I know this is a review because we did this in lab, but I just want to be really clear now because I saw a little bit of confusion in the lab. To have a dominant trait, you only need just one dominant allele, right? So if you have a dominant trait, you could be homozygous dominant or you could be heterozygous. The point is, you only need one, right? That's very important. If we're talking about phenotypes, you only need one allele to have the dominant trait. Another way of looking at it, and somehow I think this is what's confusing people more than that other concept. If you have a recessive trait, you have to be homozygous recessive, right? The only way you can have a recessive trait is if you do not have any dominant alleles. In other words, you would have to be homozygous recessive. So are there any questions about that? All right, pretty simple. Again, I'm hoping this is a review, and judging by the lack of questions, I'm hoping it is. Number 4, I haven't specifically said these words, but I'm hoping this is very obvious to you based on the way I was giving the pre-lab lecture, in lab. A sperm or egg carries only one allele for each inherited character because the two alleles for a character segregate during the production of gametes. So like I would show you an example of a mating on the board to show you how to do a Punnett square. I'd be like, okay, this heterozygote mates with this homozygous dominant person. And I said, all right, when this person goes through meiosis, half of their sperm has big T's and half of their sperm has little t's because they separate in meiosis. And when this person goes through meiosis, yes, the big T's segregate, but it doesn't matter because it's a homozygous dominant. So the only thing they have to offer is the big T. So 100% of their eggs are big T. So I said that quite a few times in lab. I hope that made sense then, and I really hope it makes sense now. So even though if you were to look at all of your cells, other than your gametes, all of your cells, you have two alleles, at least two alleles. We'll get into that later. You have at least two alleles of each gene, right? But if you were to look at any of your sperm or egg, then you would find you only have one allele because again, they segregate in meiosis. You only have one of each chromosome. Therefore, you're only going to have one of each allele. Anyway, any questions about that? All right. Good, because this is what we call the law of segregation. And this is what we were all getting. This is what I was getting to. This is the important part as far as the exam is concerned. You need to know what the law of segregation is. When the sperm and the egg unite, each contributes its alleles, restoring the paired condition of the next offspring. Let me, I want to make sure, now that I'm looking at this is a little bit of a confusing slide. So I'm going to draw a little line right here. So everything above this is what we're talking about. That is the law of segregation. So in your mind, to help you remember, you might be thinking, okay, law of segregation, obviously something is segregating. What is segregating? And the answer is this: it's the alleles that segregate. Or depending on how the question is worded, it could be the chromosomes that are segregated, right? What we're saying here is that if you look at a gamete, you only have one of each allele in that gamete. Or again, depending on how the word is questioned, or the question is worded, you only have one of each chromosome because they segregate, right? They separate from each other like they do in meiosis. So can someone tell me, if you're not thinking about biology, right, if you can somehow purge your brain of that, even though we're sitting here, what do you think of when you think of the word segregation? Anybody?
Like when you're put by yourself in jail.
Oh yeah, that's one. I didn't even think about that. Yeah, usually, yep, that is segregation. I always think of that as solitary confinement, but you're right, that is also segregation. That's another word for it. What else? What other things do you think about with the word segregation? Anybody else? At a historically black college and university, I was hoping somebody would have some sort of answer that goes back to the 60s.
When people were separated by race.
There you go. Yeah. So that's, to me anyway, when I think of segregation, maybe it's because I'm older, I don't know, but that's the first thing I think of was that dark period in our history where we segregated people by race. But nonetheless, that helped me remember as a student the law of segregation. Of course, I just had to remember, obviously, we're not talking about segregating people or doing it by race, but we are talking about segregating chromosomes or alleles. So hopefully that helps you remember that. Anyway, so yeah, I put that line up here to remind you that up above here is the law of segregation. And then that being said, we'll talk about this down here. When the sperm and the egg unite, like I said earlier, each of them contributes their alleles, and that restores the paired condition in the offspring. And this bullet point is what you already learned in chapter 8. I mean, so far, technically, you learned all this in chapter 8, just not in this context. But this is just fertilization, right? This bullet point right here is just fertilization. That's when we go from two haploid gametes, they combine to give us one diploid zygote. Anyway, are there any questions about that? Okay. Now that's a big one. Make sure you understand the law of segregation. And to truly test whether or not you understand the law of segregation, do the study guide because there's questions about the law of segregation. And if you get it wrong, I'll let you know. And then I'll, if you have questions about how to remember the right answer after you've done the study guide, then we can talk about it. All right. So here's a picture you've already seen, except now they've just thrown the alleles in there as well to illustrate this idea of segregation. So again, we have these purebred purple flowers and purebred white flowers. Obviously, if they're purebred, they're going to be homozygous. Specifically, that one's homozygous dominant. This one's homozygous recessive. When this thing goes through meiosis and produces egg or sperm, yeah, those P's segregate, right? Because that's what happens in meiosis. But it doesn't matter that they segregate, because no matter what, the only thing this purple flower has to offer is a big P. So 100% of the gametes are going to be big P. Same here, little p. That's the only thing this thing has to offer is little p, because it's homozygous dominant or recessive. So yeah, they segregate in meiosis, but it doesn't matter. 100% of their gametes are going to be little p. Now then, we have fertilization when we bring these things together. When, I don't know, maybe this is a sperm and that one's an egg. So this sperm carrying a big P comes together with this egg that's carrying a little p. And then we have ourselves a little zygote that has a big P and a little p. And then that thing grows up to a full-grown flower. And that's what it is, right? It's purple. It's big P, little p. Then this thing's also going to go through meiosis. And we're also going to look at the law of segregation again. But this time it's a little bit more apparent, because this time there are actually different alleles. So it goes through meiosis. Literally half of its gametes are going to be big P. Half of its gametes are going to be little p. So then that's where we get the Punnett square, right? So if, each parent has this situation, big P, little p. So big P, little p for one parent, big P, little p for the other. And then you look at the possible combinations when you bring two heterozygotes together. You have one homozygous dominant, one homozygous recessive, and two heterozygotes. Which brings me to another point. I mean, we just illustrated the law of segregation, so that's good. But another thing you should recognize for the exam is when you see this ratio right here, forget the letters even, when you see the genotype ratio of 1 to 2 to 1, you should know that I'm talking about a monohybrid cross. And that'll make more sense a little bit later when I introduce you to something called a dihybrid cross and give you a different ratio. And you'll see the ratios are so much, they're really different. So it should be easy to remember once you see both of them.
And you said when we see the 1 to 2 to 1, that is what?
The monohybrid cross, which is all this what we're talking about. And to remind you, because I used the word monohybrid cross today, but I didn't say what it was. So here, the monohybrid cross means we're crossing these flowers, but we're only looking at one character, right? We're just looking at flower color. That's what the mono part means, just one. Now later, we're going to talk about things where we're looking at two things. Like it could be flower color and pea color, right? When you're looking at two things, as you're going to learn later, that's called a dihybrid cross. When you're only looking at one thing, it's a monohybrid cross. It's a good question. All right. Punnett square. I'm going to go through this very quickly because we've already talked about it and you've already done it in lab, but it highlights the four possible combination of gametes, the four possible offspring, and each square represents an equally probable product of fertilization. Yeah, I didn't have anything on the next. Oh yeah, see the previous slide. Yeah, that's the Punnett square right there. So what I will do between now and when we have the exam is I'll come up with some problems, hopefully, and I'll give them to you as extra credit or independent work, whatever, where you can do some Punnett squares. And I might ask you a question if a person with this color eyes mates with a person with that color eyes, what are the chances that their kids are going to be blue-eyed, or what are the chances their kids are going to be brown-eyed, something like that. And you can get practice because you should have at least one question like that on the exam. And I'll probably have a lot more questions like that on the exam for extra credit. Because really, it's kind of practical, not necessarily for eyes, but later on, you're going to learn that you can do these things for heritable diseases, for example. So it might be nice to know what are the chances of you and your mate having a kid with some disease that runs in your family or your mate's family. So anyway, any questions about this slide? All right. So we are done talking about Punnett squares. Let's talk about genetic alleles and homologous chromosomes and how they are connected. And hopefully at this point, I've already given you all this information in separate lectures, but now I'm going to bring it together. So hopefully you kind of already know this. And what I'm about to tell you, you're like, oh, yeah, I knew that. I just didn't think of those things together. So here we go. Some stuff you do definitely already know, the relationship between the words phenotype and a genotype. Your book in this portion of the chapter describes your phenotype as a physical appearance. I should point out that it's not always an appearance. Sometimes it's something you can't see. Like it could be a disease. A disease could be a phenotype, and some diseases you cannot see. So anyway, your genetic makeup is your genotype. In other words, are you homozygous dominant? Are you homozygous recessive? Are you heterozygous, right? That's your genetic makeup. So in my opinion, the better way to describe genotype and phenotype is your genotype is your genetic makeup, like we said, right? Homozygous dominant, heterozygous, homozygous recessive. And your phenotype is the result of your genotype. I like to say the result of your genotype better than your physical appearance because, again, not all of it is the physical appearance. Anyway, each character, now we're going back to talking about Mendel's experiments with the peas, the pea plants. Each character had the same inheritance pattern, which is where the parental trait disappeared in the F1 generation and reappeared in 1/4 of the F2 generation. So again, remember we had the P generation that had, this is horrible, I can't really draw it, 2 flowers, one of which was purple, one of which was white. Those two crossed and had offspring, and that offspring is what we call the F1 generation, right? We had that purple flower, and that's what this says right here. The parental trait disappeared in the F1 generation, meaning all the flowers were purple, right? Somehow that white trait disappeared. But then the F1 generation was crossed with themselves, and that gave us the F2 generation. And he found in the F2 generation, I wish this is so horrible. I need better tools. But anyway, he found in the F2 generation that about 1/4 of the offspring had that lost trait. So again, this is everything I've already told you. I'm just reiterating it. Are there any questions? All right, the next slide I'm going to show you shows a pair of homologous chromosomes, and you should be familiar with that from chapter 8. But just as a reminder, a homologous chromosome is a chromosome that has the same alleles at the same place. Speaking of the same place, there's a word for that, and I'm not sure if I've officially given you this word or not. I know I think I've unofficially threw it out there in lab. The gene locus. That is a specific location of a gene on a chromosome. And I've used that concept a lot, even if I didn't use the term, because I know I've given some off-the-wall examples of, like, I don't know, the gene for bad breath is found on the tip of chromosome number 7 or something like that, right? If I'm talking, so when I do that hypothetical situation, when I discuss that hypothetical situation, what I mean when I say it's at the tip of chromosome 7, what I've given you is the locus. It's the location. You can almost think of it as the address of a gene. And mostly for all organisms of the same species, they all have the same address, the same locus. So the gene for, chin dimple, I don't know where it is, but wherever it is on my chromosomes, it's going to be there on your chromosomes too, and every other human on Earth who doesn't have anything wrong with them, with their chromosomes. So anyway, that's what a gene locus is. And again, the alleles of a gene reside at the same locus on homologous chromosomes. That's basically, I'm just repeating myself in a different way, and this will make a lot more sense on the next slide when I show you pictures. And of course, as we already know, when we're talking about these alleles, they might be the same, they might be different. In other words, they might be homozygous, they might be heterozygous. You already know this, but here's a picture. So there's a homologous chromosome we're looking at here. If we're talking about humans, this could be number 1. It could be the sex chromosomes. It could be number 22, 17, anywhere in between. Doesn't matter. The point is we're looking at a pair of homologous chromosomes. And let's just say randomly, and we don't know this, but let's just say this is the one you got from your mom. This is the one you got from your dad. And let's say B is the gene for bad breath. And if you look at this part of the chromosome, you will find the gene for bad breath, whether it's from your mom's chromosome, the chromosome you got from your mom, or the chromosome you got from your dad, or any other human being's chromosome. If you were to look, right there would be the gene for bad breath. And of course, I'm making that up, the bad breath part, but that is how it works. It's always at the same location, the same locus. And then here, again, I don't know, same thing, right? So the point here is when this is pointing here, that's just saying, all right, this is the gene loci. This is the gene's location. This is the gene's address, if you will. And then, of course, this just shows you what you already know. Here we have somebody who's heterozygous because we have big B, little b. Here we have somebody who's homozygous. This is all the same person, but here this person is homozygous recessive when it comes to that gene. And when it comes to this gene, this person is homozygous dominant. Any questions about this picture? All right. Just to be clear, because I remember being a freshman and looking at a picture very similar to this, and it confused me when I looked at it and I wasn't in class. But I was like, wait a minute, this doesn't make any sense. Because these, this says gene loci and it points to these three things. But then it points to allele and points to the same thing. So remember, just to be clear, yes, these are the same things, but when we're talking about allele, we're just talking about two different versions of a gene, right? That's all. We're just talking about the different versions of the gene. And what this is saying is this is the gene's address, if you will, right? So the gene B is located towards the tip of this chromosome. Gene P is located more towards the centromere of the chromosome. And gene A is located between those two. That's all it means. All right. So here's where it gets a little bit trickier. In my opinion, when you first get introduced to this idea of the law of segregation, it's pretty simple, right? The chromosomes segregate in meiosis. Pretty simple. The hard part now is when I introduce you to this new concept and then remembering the difference between those two concepts when you have an exam. The next word for attendance, I'm not going to say. I'm going to show you. I'm going to circle it right now. That is the next word for attendance. So make sure you can see that. If you're just listening with your eyes closed and you're relaxing, which is okay, briefly open your eyes and take a look at the screen. This is the next independent work. Excuse me. What the heck? I can't think this morning. This is the next word for attendance. Anyway, like I mentioned earlier, I think it was Cordone that asked the great question about the ratios. And I was saying, yes, if it's a 1 to 2 to 1 ratio, you should recognize that as a monohybrid cross. And when I said that, that reminded me to tell you there's also something called a dihybrid cross. So here we go. Let's talk about a dihybrid cross. It's basically the same as a monohybrid cross, except instead of looking at one character, we're looking at two characters. It's pretty simple. Biology and science in general just has a tendency of giving fancy, hard to remember words or names to just simple concepts, right? Monohybrid cross, you could have just said, yeah, we're crossing things and we're looking at one character, but instead they made a word for it. Same thing with this. Instead of saying, yeah, we're crossing things and looking at two characters, someone thought, well, let's give that a name that's going to be hard for students to remember. And that name is dihybrid cross. But here's where it gets a little bit more complicated. If the two characters were inherited together, which they're not. So again, we're looking, all right, let's think about the lab, right? The lab we did. Chin dimple was one thing and interlocking fingers was another. Now remember, chin dimples, oh no, maybe it's a bad example. No, that's a good example. In both classes, the chin dimples was overwhelmingly recessive. Not many people had a dominant version. This is not a perfect example that I'm about to give, but it's an okay example. It's a good way to start things off. Anyway, no, I don't even want to get into that. That might make it more confusing. Anyway, if two genes were inherited together, which they're not, let's talk about what would happen. And as I'm describing this, keep in mind, this is a hypothetical situation that is not true. If they were inherited together, then the F1 would produce only the same two kinds of gametes they received from the parents, and the F2 would still show a 3 to 1 phenotypic ratio. And this is a really long, hard, complicated way of saying if the genes for the two characters were inherited together, and you could just paraphrase this and say, then things would be exactly the same way as they were with a monohybrid cross. That's all that's saying, right? But they're not. What really happens is this. The two characters are actually sorted independently, and this is where we're getting at this idea of independent assortment. And because of that, the F1 produced 4 genotypes, and we are using R's as an example. It doesn't always have to be R, but you could have big R, big Y. And again, these are 2 different genes, right? Not even alleles, but genes. And this will make a lot more sense when I show you the picture. Big R, big Y, little r, big R, little y, and of course, little r, little y. And these would all be expressed equally. And here's the important part for the exam, and I'm going to come back to all this on the next slide. It'll make a lot more sense on the next slide. But like I said, when you see a 1 to 2 to 1 ratio, like I showed you on that other side, you should recognize that as a monohybrid cross. When you see a 9 to 3 to 3 to 1 ratio, you should recognize that as a dihybrid cross. Now then, let's get to it where I can show you a picture and it should make more sense.
Now, will those always be the numbers, or?
Great question.
Just...
There's a portion in this chapter where we talk about where those are not the numbers, but for the sake of the exam, when you see those numbers, then they are. Or here's an even better way of putting it. Let me ask you this: how many numbers? Forget that that's a 9 and that's a 3 and that's a 1. But how many numbers are in that ratio?
4.
Perfect. 4.
The other one had 3.
Exactly. Okay. So that was a great question, Cordone. And I don't even get into the fact, I don't even talk about too much about how they could be different because it's very complicated. But the one thing that is definitely the part that doesn't change no matter what is if the ratio has 4 numbers in it, that is a dihybrid cross. And if the ratio has 3 numbers in it, it is a monohybrid cross. I'm so glad you asked that. And I've never really thought to teach it this way. But now that I'm thinking about it, obviously dihybrid versus monohybrid, 2 is bigger than 1, right? And to help you remember, 4 is bigger than 3. And with this multiple choice exam, I'm never going to try to trick you. So there won't be any ratios that have like 5 or that have 2, right? If we're talking about genotypes, and dihybrid cross versus monohybrid cross, they'll either have 4 or they'll have 3. All right, this is a little bit complicated, but it's very important. We're really looking at the independent assortment here. So yes, we're only looking at a P here, right? We're looking at the P. We're no longer looking at the flower, but we are still looking at 2 different characters. In this case, even though it's just the P, we're looking at the color of the P and we're looking at the shape of the P. Now, this color or this P is yellow and it's smooth. And it just so happens that yellow, right here, is the dominant trait, right? It's expressed by a big R or a big Y. And it's, I don't want to say smooth. It is smooth, but round is a better way of putting it. So it is round, right? That's that. So it's homozygous dominant for being round. It's homozygous dominant for being yellow. This one is homozygous recessive. So it's not round. It's wrinkled, right? So it has the recessive version. And it's not yellow. It's green. It has the recessive version. Now, if these things were segregated the same, like if they were on the same chromosome, then you would expect every time you had a round one that it would be yellow. And every time you had a green one, it would be wrinkled, right? That is what you would expect. And you would get a 3 to 1 ratio if that were the case. But they don't segregate the same, right? They are completely different alleles, possibly on completely different chromosomes. So they have nothing to do with each other. So what really happens is, I wish your book did a better job of showing this. Man, there's got to be a better way of showing this. Yeah, I guess there kind of is. So when this thing goes through meiosis, yes, that big R, those big R's segregate. And yes, those big Y's segregate, right? That's really hard for me to draw that. Yes, they do. But because the only thing this offers is big R and big Y, then 100% of their gametes are going to be big R and big Y, no matter what, right? Even though, yeah, no matter what, they'll all be big R, big Y, because that's all this thing has to offer. Likewise, this thing too. Yeah, those R's are going to segregate in meiosis, and those Y's are going to segregate in meiosis, but that's the only thing it has to offer is a little R and a little Y. So 100% of the gametes are going to be that. Therefore, 100% of the F1 generation will definitely have this heterozygous, 2 heterozygous, right? You have the big R, little r, and you have big R or big Y, little y, right? Those are two things that are completely separate from each other. One's for the shape of the P, one's for the color of the P. Now, here's where it gets a little bit more complicated. Because these things are not on the same chromosome, they have nothing to do with each other, meaning big R is going to do its own thing, or excuse me, R's are going to do its own thing, and Y's are going to do their own thing. So here's all the possibilities here. You could have a big R and a big Y get together. Here, let me erase this. This might make it easier. There we go. Come on, technology. No, not a laser pointer. I'm sorry, guys. So again, you could have a big R and a big Y in the sperm, or you could have a big R and a little Y in the sperm, or you could have a little R and a big Y in the sperm, or you could have a little R and a little Y in the sperm. And that's what this shows right here. This is really complicated. What I want to do after this is, because I can't spend too much time trying to explain it, sometime this weekend, I'll either come up with my own video and like really go into depth on this, or I'll find one that's already out there on the internet and share it with you guys. But the point here is, again, these things assort independently of each other, hence the name independent assortment. Therefore, you have this 9 to 3 to 3 to 1 ratio, and they have nothing to do with each other, right? They're on completely different chromosomes. Therefore, they're not inherited together.
Somebody's hand is raised. I don't know if they meant to do it, but.
Oh, thanks. All right. So whoever has their hand raised, did you have a question? All right. Maybe not. If you think of it, let me know. So that dihybrid cross supported Mendel's hypothesis that each pair of alleles segregates independently of other pairs during gamete formation. Therefore, the inheritance of one character has no effect on the inheritance of another. Here, let me give you this example really quickly. I'm going to do another one of these wacky examples that don't really exist in nature, but that's okay. Here's a chromosome. All right, let's call it chromosome number 3. I don't know. The number really doesn't matter, but let's just say that's chromosome number 3. And this is the one you got from your mom. And then, of course, you also have a version from your dad, right? Chromosome number 3. And let's say that from your mom, there's a line here. So let's use these as the alleles. Why not? Because the line's already accidentally here. All right, let's say there's a gene that controls your breath, right? And then it's located right here on the chromosome. And let's say your mom has big B for bad breath. Your mom has bad breath. And your dad, meanwhile, has little b because he was homozygous recessive. He had good breath, little b, little b. All right, so that's that gene, right? Bad breath, the gene breath. And then up here on the same chromosome, on the same chromosome, at this spot on the chromosome, there was a gene for back hair. And it just so happens your mom's not very attractive. She's got bad breath, and she's got a hairy back, big H. Your dad, meanwhile, has good breath and a bald back, right? Now, these things are on the same chromosome. So if they were on the same chromosome, you would imagine then when it comes to getting inheritance, right, you're either going to get your mom, you're only going to get, how do I put this? Let me put it a better way. When you produce sperm or egg, right, when you make your gametes, you're either going to give them this mom chromosome or this dad chromosome. You guys already know that. And what that would mean if they were on the same chromosome, then if they got your mom's chromosome, that means if they would get your mom's bad breath allele, that means they would also get her hairy back allele. Does that make sense? See how it's on the same chromosome? Can you guys follow me so far? When you make sperm in this situation, when you make sperm or egg, then your sperm or your gametes are either going to have bad breath and a hairy back, or they're going to have good breath and a not hairy back, right? Because you're getting one or the other chromosome. Now, here's a more realistic situation. And this is what we're getting at with the idea of independent assortment. I guess I could have kept that. Whatever. Okay. So here you have, again, let's just say, good grief, technology. let's just say again, hypothetically, we're talking about chromosome number 3, and we're looking at the bad breath thing. Now, let's say that hairy back gene was not located on the same chromosome. Let's say that was located on, I don't know. Good grief. This is killing me. Anyway, ignore that long line. That was a mistake. Let's just say we're looking at chromosome number 7 for this. All right. We're looking at chromosome number 7, horribly drawn.
Oh, look at the 21-year-old.
Okay. So there's chromosome number 7. All right. Number 3, number 7. Number 3's got the chromosome for bad breath. Number 7, let's say this is it for back hair. Again, there's your chromosome from your mom. Here's your chromosome from your dad. And again, your dad he's recessive. He gave you the recessive gene. He gave you the non-hairy back gene. Your mom, on the other hand, being the bad breath and unattractive person she is, gave you a hairy back gene. Now then, here's what we're getting at when it comes to the Mendel's law of independent assortment. The bad breath gene is on chromosome number 3. The hairy back gene is on chromosome number 7. Now, when you go through meiosis and produce sperm or egg, remember what I told you about the whole 8 was 8 trillion, 8 million? 8 million different possibilities of ways that the chromosomes can line up. Remember, it's not all, when all your chromosomes line up in meiosis, it's not, I'm going to only draw 3 pairs. And remember, you have 23 of these. I'm only drawing 3. Your chromosomes line up in the middle, your homologous chromosomes. And it's not like all of them are mom on one side and dad on the other. It's a good time to remind you of that. Remember, they line up independently of each other. It could be all mom on one side and could be all dad on the other side. Or this could be mom and dad. This could also be mom and dad. And then here you could have dad and mom, right? They line up independently of each other. Therefore, when you're producing gametes, you could give your kid the bad breath allele and the hairy back allele, right? Or, because you could give them both chromosomes from your mom, or you could give them the bad breath allele and the bald back allele, meaning you could give them a mom chromosome and a dad chromosome. Or you could give them 2 dad chromosomes, in which case it would be good breath and a hairy back. Or you could give it a dad chromosome for number 3 and a mom chromosome for number 7, meaning it would be, good breath, but a hairy back. That's what we mean by independent assortment. These things, as you know from the last chapter, line up independently each other. All 23, when they line up for meiosis, metaphase and meiosis 1, there's no rhyme or reason. There's no rules. It's not all mom on one side and dad on the other. They line up independently of each other. Therefore, the inheritance of these things, the bad breath and the hairy back, they are not related to each other because they're on completely different chromosomes. That's all that means. Woo! Your next word for attendance is going to be black dog. Which is not just what's on the screen, but it's also a great song by Led Zeppelin. If you don't know it, feel free to look it up. Anyway, so your book's giving this example of, let me back up, sorry. We're still talking about independent assortment. Oh, yeah. And we're looking at 2 different characters in dogs. We're looking at their coat color and their ability to see. Having a black coat is a dominant trait, and being able to see is a dominant trait. So we know if a dog is black and can see, or a Labrador, if a Labrador is black and can see, we know that it has the dominant phenotypes for both. Therefore, we know its genotype has at least one dominant B, one big B and one big N, right? Doesn't even matter what the other one is. Could be big B, could be big N, could be little b, could be little n. Doesn't matter because, again, this is the dominant trait. So all you need is one dominant allele. Now here we have a black dog that's blind. Therefore, we know because it has the dominant color phenotype that, again, it would be big B blank, right? Because it doesn't even matter. Could be big B, little b, doesn't matter, dominant phenotype. But we know if it's blind, it must have the homozygous recessive genotype. It has to, because blind is the phenotype, right? That's the recessive phenotype. Therefore, you would have to have homozygous recessive genotype. Then the next possibility, we have this dog that has the recessive hair phenotype, which is chocolate, but the dominant vision phenotype, which is the fact that it can see. So again, because it has a recessive hair phenotype, we would know that its genotype is homozygous recessive. And because it has the dominant vision, we know that it could be big N blank or big N, big N or big N little n. As a matter of fact, now that I'm thinking about it, and this doesn't help you guys, but from now on for my future labs, that part where you had to write, for chin dimple, you had to write big D, and then the word or, and big D, little d. I might just have you guys write big D and then blank, because that's what your book does. So anyway, and then finally we'll move to the black dog. This dog has both recessive phenotypes. Therefore, we know it has both recessive genotypes. And this is what we're working with. I'm not going to ask you if you have any questions yet, because I'm just setting this up. And we can look at the possibilities of these dogs mating. This is the mating of double heterozygotes, meaning, even though none of those are shown, let's just assume it would be this. Let's just assume we are mating a big B, little b. This is so horrible. With a big B, and a big N, little n. Let's assume we're mating two dogs that have this genotype: big B, little b, big N, little n. Two of them. That is what we mean by mating of a double heterozygotes. Now, if you were to mate those dogs as many times as it would take to get this many puppies, this is about the same ratio that you should expect in that situation. You would get about 9 of them that are black with normal vision. You should have 3 of them that are black that are blind, about 3 of them that are brown with normal vision, and about 1 that is blind and chocolate colored. So again, there's that 9 to 3 to 3 to 1 ratio that you should expect with this dihybrid cross. And really, that's complicated. The most important thing you understand from this picture is the fact that the gene for vision and the gene for coat color have nothing to do with each other, right? They are on completely different chromosomes, which is why you see this ratio. That's important. That's the most important thing. Don't forget the ratio, but more important than what the ratio is what it indicates, which is the fact that these things are independently assorted, right? They do not line up together on the same side in meiosis, in metaphase of meiosis. That's all that means. All right. Any questions about that? And in my opinion, this is probably the differences between independent assortment and segregation are probably the hardest things in this chapter, if not this exam. So I will, like I said, try to find time this weekend to find some more videos to share with you. And maybe even come up with some questions so you can get some points for watching them, because that is, again, a very important concept. When we get back on Monday, we can use this concept of, well, you'll see. And basically, you're going to learn how to do a test cross to determine an unknown genotype. And what do I mean by an unknown genotype? Like this, right? We don't know if that thing is big B or big B, little b. All we know is it has a dominant phenotype, so it could be either one. Same thing with that. But we'll do that on Monday. Are there any questions before we stop recording? All right. And remember, like I said earlier, I will be online for office hours, but I'll be done early at 9:50 because I have a biology department meeting to go to. And then after that, probably around 11:30, you should be able to reach me by calling my office number, because that is forwarded to my cell phone number. And that's all I have. You guys have a great spring break. And remember that even though you are on spring break, I am not. So I am here to help you if you want it.
You too.
Thank you.